Okay, great. So let's get started, everyone. Hi, I wanted to start by thanking everyone so much for joining us today for LARVOL's live discussion on the top abstracts coming out of ASCO GI. My name is Kanisha Gala. I'm an oncology project owner at LARVOL, and I'll be starting us off by introducing our panel. So today's moderator is Dr. Shalaan Beg. He's the Director of GI Medical Oncology at UT Southwestern Medical Center. He's also the Medical Director of the Clinical Research Office at Harold Simmons Cancer Center, and he's an Associate Professor of Internal Medicine at UT Southwestern. Our panelists are Doctors Rachna Shroff, Rachna Vijayvergia, and Kevin Knopf. Dr. Shroff is Chief of GI Medical Oncology at the University of Arizona. She also serves as the Director of both the University of Arizona Cancer Center Clinical Trials Office, as well as the Arizona Clinical Trials Network. Dr. Vijayvergia is Assistant Chief of GI Medical Oncology at Fox Chase Cancer Center. She serves as the Medical Director of the Partners Program and is an Assistant Professor of Hematology Oncology at Fox Chase. And Dr. Knopf is Chief of Hematology Oncology at Highland Hospital and an Assistant Professor of Clinical Medicine at UCSF. In preparation for today's live discussion, we have asked our panelists to submit their list of top abstracts coming from ASCO GI. We then consolidated that list, and we will now be discussing the top 4 abstracts during this live discussion. We've specifically asked our panelists to comment on how these top abstracts will affect standard of care in GI oncology, and I'm very much looking forward to this discussion. So with that, I will hand over the reins to Dr. Beg. And again, I really want to thank everyone for joining us.
Great. Thank you very much, Dr. Gala. So I want to thank the organizers at LARVOL for setting up this webinar and for giving us an opportunity to share our thoughts with each other and with you. And I'd like our panelists to imagine that we're in a fancy hotel in San Francisco with a fantastic view of the bay, and we're sitting on a plush couch. And being the science geeks that we are, going over the discoveries of or the research that's been presented in the last 3 days. We have some recent advances in cholangiocarcinoma, hepatocellular carcinoma, and pancreatic cancer, which we'll be discussing today. So let's dive right in. So if we can have the next slide, please. All right, so this GI ASCO, we had final results of a phase 2 study of infigratinib, which is an FGFR selective tyrosine kinase inhibitor for previously treated cholangiocarcinoma patients who harbored FGFR2 gene fusion or rearrangements. So next slide, please. This was a clinical trial taking patients who either progressed or were intolerant to gemcitabine-based chemotherapy and offered them infigratinib. The primary endpoint was overall survival. They enrolled about 108 patients. Median age was 53, predominantly female, 62% female, 78% white, good performance status patients, and most of them had stage 4 disease on progression. The panel on the bottom left shows the response rate waterfall plot. The overall response rate was fairly impressive. 32%. They had 1 complete response as well. And then on the bottom right, really to highlight the toxicities and the side effects that we see with this class of medication, since it is a relatively new class of medication for our patients with cholangiocarcinoma and cancer patients in general. We're seeing a new class of toxicity emerge, primarily stemming from the mechanism of action with hyperphosphatemia and stomatitis and some eye irregularities. So I wanted to invite Dr. Knopf to comment on his thoughts on the abstract. There were other studies that were presented with IDH for IDH mutated colon cancer patients as well. So Dr. Knopf, what was your take on the cholangiocarcinoma studies that were presented at the GI symposium, particularly this one with FGFR?
It's certainly become more interesting for treating cholangiocarcinomas over the past 20 years. In general oncology practice, they're not particularly common, but we see a fair number of them, and of course they're always locally advanced or metastatic at the time we see them. And the FGFR inhibitors have gone from being part of the growth factor pathway that we learned about in medical school and residency to being a target, which has really changed things. And for me, the first FGFR inhibitor I used was in a bladder cancer patient, surprisingly. So it's nice to see this show up in cholangiocarcinoma as a target. What will change a lot, I think, maybe in the past year or two with cholangiocarcinoma is that we're used to treating it primarily with cytotoxic chemotherapy. For a while, we were using 5-FU and gemcitabine, and then we had good data on first-line gem-cis, which I think has become the standard of care off trial. Earlier, we had tried to add Avastin for pancreas cancer, which is a similar histology without much benefit. But now we have to bring in targeted therapies to the world of cholangiocarcinoma. So in this GI ASCO, the interesting abstracts I thought were related to this FGFR inhibitor trial and also the IDH inhibitor trial, and how you're going to start approaching cholangiocarcinoma from a molecular standpoint. The need to sequence or study these molecular targets another way, which hadn't been part of our toolkit or necessity of practice maybe 2 years ago. And then actually the molecular biology of this is particularly interesting because there's another FGFR target, which is a monoclonal antibody. This medicine is an oral tyrosine kinase inhibitor specifically to FGFR mutations that are fusions or rearrangements. Now, in this study, the patients were relatively young and relatively good performance status, which may limit its applicability for all cholangiocarcinoma patients. The median number of regimens they could have in the study was closer to 2 than 1, although there was a hint that patients had a higher response rate with run prior. So this may replace second-line attempts at FOLFOX or modifications of that with 5-FU alone after GemCis. It will be really interesting, I think, for this drug to be developed more in phase 3 trials and see what the overall survival differences are as other molecules are competing for this. So the interesting things, I don't know the FGFR molecule that well, and I'll have to go and read the molecular biology of it. In this study, they had about 8, maybe 9 different fusions, and one of them was present in 35% of patients. But being a small study, they didn't really comment on whether one fusion or rearrangement was more likely to respond to the medicine than another. So that's an interesting thing to comment on. It'll be interesting as they're probably doing the phase 3 trials or heading that way, how this will sequence in terms of the with GemCis or after GemCis, and patient selection for the phase 3 trials. It's a fairly straightforward medicine to take. It was a pill, 125 milligrams, 3 weeks on, 1 week off, and we're all used to using tyrosine kinase inhibitors for other malignancies. The patients were fairly fit. It was a standard phase 2 approach in about 120 patients. And most of the patients, when they took this, their response was stable disease, which is actually a very good marker for cholangiocarcinoma that's refractory to first-line cytotoxic chemotherapy, with a reasonable partial response rate. No complete responses, which is not unexpected for this disease, which has been notoriously hard to treat. I think the toxicity is reasonable, although for oncologists in general, we're not used to dealing with ophthalmologic toxicities, so that may require us reading a little bit more about that, or finding an ophthalmologist we can talk to every once in a while. 20 years ago, we never really had to worry much about rashes, and now we have to learn all sorts of things about rashes from using PD-1 inhibitors, and we didn't have to know much endocrinology day to day, but now we have to understand all that when we're giving PD-1 inhibitors in GI cancers, and ipilimumab causes its own set of toxicities. So I think in a way it's frustrating, obviously, but it's also very nice for general oncologists and GI oncologists to have to know more internal medicine to manage these patients day to day. So the ophthalmologic toxicities, some of them sounded a little frightening to me. Retinal toxicities seem like more of a worry than dry eye, and there's actually a lot of medicines being developed for dry eye. But I would need to see what those retinal toxicities were. Fortunately, most of those were grade 1 toxicities. And then there was arrangements of calcium and phosphate with this, and I'd have to learn a little bit more about the hyperphosphatemia and how often that has to be monitored to do this drug. So not a particularly toxic drug, as again, most of these were grade 1 toxicities, not really grade 3 or grade 4. Certainly looks like a very promising molecule, either after GemCis or depending on how the phase 3 trials are done with GemCis, and brings also, I guess, to the forefront the need for molecular marketing, molecular assessment of biliary cancers, which we haven't really done in pancreas or cholangiocarcinomas before. So we've done this a little bit more with colorectal cancers, where we had to determine KRAS status for cetuximab use, and now HER2 for upper GI cancers has become more. So in my practice, we're not routinely doing next generation sequencing for GI cancers, but we'll have to think about our molecular approach to these cancers as these targeted molecules come to the clinic. This one is away from being to the general clinic, but certainly I think academic centers are going to want to have this on clinical trial moving forward, given sort of how poorly we do with cholangiocarcinoma overall. So I think it's a promising molecule. The results were reasonable for a phase 2 trial with 120 patients. The toxicity is a little unique, but nothing I think that a medical oncologist can't handle by reviewing a little bit of the medical textbooks, and represents a phase forward for cholangiocarcinoma. The questions I had after seeing this presentation, in addition to just some of the specifics about the trial, is how this target FGFR is on other tumors. So I'm going to have to go back to my textbook on Monday. I know it's in bladder cancer, but I don't know what other malignancies it's in, and whether this particular molecule would have broader applications than just cholangiocarcinoma. In particular for GI cancers, is it present on pancreas cancer? Is it present on cancers of the ampulla of Vater? So I'd actually like the other panelists to sort of comment on what they know about FGFR mutations and other GI cancers. And then the other question that struck me is, I think we're going to talk about the IDH1 target. How do we sequence these if they become present in the clinic? How do you, in particular, look at FGFR, and is there certain rearrangements that are more likely to respond to this or not, which we probably didn't get a signal of with this phase 2 trial, as it only had 120 patients with a lot of different mutations. But are we going to have to take our molecular assessment of cholangiocarcinoma down to that level of which fusion is actually involved in order to hit the target? And then how do we trade off FGFR versus IDH1 targets with these malignancies? We don't typically see these patients for adjuvant chemotherapy the way we do pancreas cancer, just because it's less prevalent than pancreas cancer. But for busy GI oncology practices, that would be another question: is do these molecules have any role in adjuvant patients to increase the cure rate, which is not so great even for stage 1 patients. So all in all, I think one of the more interesting developments in cholangiocarcinoma over the past couple of years.
Great, thank you very much. So we weren't planning to officially, I didn't put any slide together for the IDH study, but Dr. Shroff, you were the discussant, so you spent quite a while over the last couple of months thinking about FGFR and IDH1 in preparation for your talk, I'm sure, this afternoon. How do you see these treatments factoring into the sequencing for treatments for cholangiocarcinoma patients? Dr. Knopf talked about challenges in molecular profiling, talked about learning new skills in managing toxicity. So what has your experience been, and what do you see in the horizon for cholangiocarcinoma treatments if you look at these initial few studies that are reporting out?
Yeah, I mean, that was a great summary, Dr. Knopf. I agree. I think that I said this in my discussion, I think that the time is now. At this point in time, we should be sequencing every single cholangiocarcinoma patient between IDH, which is 20%, FGFR2, which is 15%, BRAF, which is 5-7%, and then like a smattering of things like patients and such. It's about 40 to 50% of cholangios that we can do something with in terms of targeted therapies. Pemigatinib, we know, is already FDA approved, so we do have an FGFR inhibitor available to us for FGFR2 fusion positive cholangiocarcinoma. So it's not just if we have a trial, you know, and then the FDA has granted priority review to infigratinib based on this phase 2 data. So we may have 2 very soon. So I think that it's really important that we look for these FGFR2 fusions. Speaking to the question about the partners, so we now have over 300 patients worth of data between the futibatinib data, the pemigatinib data, and the infigratinib data. Partner agnostic, the response rates do not matter what the partner is. So 35% in the infigratinib had partners that were completely different than the BIC1 and the more common partners that we see. The Insight group has published the pemigatinib partners as well. So there's no correlation between response and specific partners. So it's just as long as you have an FGFR2 fusion, it does not matter what the partner is. It's an early oncogenic driver and it responds. And the response rates are all pretty similar. I think the response rates are better in the earlier lines of therapy. So second line is better than third line. And that's, I think, what the infigratinib data shows us for sure. Pemigatinib was more early pre-treated patients. They were more second line patients. But I agree. I mean, I think, you know, where do we put this in the context? The question about IDH and FGFR2, I mean, those are, we think, mutually exclusive. So we don't usually see FGFR2 fusions and IDH mutations. So we don't have to think about, do we give IVO or do we give infigratinib? But, you know, my general sentiment is that if you find a fusion or you find an IDH mutation, we should give them the targeted therapy as soon as we can after gemcitabine-based therapies. I don't think that we have data that says we should give it yet in the front line. The frontline study that's ongoing is comparing gemcis to infigratinib. So it would not be, I think it would be very hard to add FGFR inhibitors to gemcis. You know, while we're getting better at managing the AEs, I think your points are very well taken, Dr. Knopf. There's a lot of complexities to the on-target effects of these FGFR inhibitors. You know, the phosphorus, to be honest, in clinic is not, I mean, other than the fact that the protocol mandated that we treated these patients with, you know, phosphate binders and things like that, it was not clinically significant. It was just a number that the protocol defined us having to treat, which was really frustrating because I don't think that there's a dramatic clinical impact, but the stomatitis, the dry eyes, the nail bed changes, I mean, there's absolutely cumulative toxicities, and so I think it would be hard to add it to gemcis. Because all the companies very wisely in their frontline studies have looked, it's futibatinib versus gemcis, it's pemigatinib versus gemcis, and it's infigratinib versus gemcis, which unfortunately is going to lead to a lot of other questions. But so yeah, so I mean, I think that it's definitely it's time to doing sequencing, and I think that the era of just treating with gemcis and FOLFOX is hopefully over. I mean, obviously there are people that don't have targets and we still are using those drugs, but I definitely think that there's a role. Now, I mean, the IVA, I know we're not discussing the IVA data as much, but you know, the ivosidenib data, that's the other question is it going to be FDA approved? Are we going to get it available to patients? Which I think remains to be seen.
Do you have any sense of whether the IDH1 mutation inhibitor is more efficacious than the FGFR inhibitor? I mean, I don't know which patients have both mutations present or not because we don't have
because they're exclusive, right? But I mean, what I'll say is not that you should do this apples and oranges comparison, but in terms of like going to your question about adjuvant, there's a lot of interest in creating perioperative trials right now. Myself and Dr. Maithal are writing one, and then the NCTN mechanism is looking at one, using FGFR inhibitors kind of neoadjuvant to downstage, because they do have response rates in the 30% range, and perhaps if you use them earlier in resectable disease, maybe it would be even better. But you know, at the end of the day, in the IVO data, the response rate was 5%. So it's much more of a cytostatic drug. So I don't know how much value it would have in peri-op. But there is some interest in maybe thinking about it in an adjuvant and that sort of thing. So I think there's a lot of different ways that people are thinking about how to develop the drugs. I just, it remains to be seen how we're going to make it part of our treatment.
Even in the metastatic setting, with so many players in the market about to come in, the question begets, right? What is the mechanism of resistance and how we can, once they progress on one, so like, there's some data about futibatinib and how that could actually be effective, or TAS-120 in patients who have progressed on a prior FGFR inhibitor, and how it could be better. So like thinking of ideas why you could have, progress on one FGFR, just you do with the, T790M mutation in lung cancer, osimertinib, something like that would eventually evolve for cholangiocarcinoma.
Yeah, I definitely agree. I mean, I do this in clinic. I can tell you that, like, because there's expanded access for futibatinib, I have absolutely, based on the gatekeeper mutation profile and what I get on ctDNA, if they have, the specific mutations that we think change the binding domain in a very specific way, and that's why they developed resistance to PEMI or to infigratinib, then I give them the covalently bound futibatinib, and they respond again. So it's great. I mean, it is. I totally agree. I think the question is how many are we going to have available to us, and how can we sequence them, and how can we strategically figure out to use all of them?
Right. What sort of mutational milieu are you seeing with your pancreas cancer patients? I mean, these are being tested in cholangio, but the epithelium is very similar. Is it generalizable or
so
no?
I know that because I recently wrote like a LOI for a study for using one of these drugs in all GI cancers.
Right.
And I know it's like 4% for pancreas, 4% for colorectal, about 6 to 10% for the gastric. And that's why we saw the gastric study with BEMA earlier during the GI ASCO as well. So gastric cancer, yes, we have a study that shows it's a target where they were actually successfully able to combine it with frontline chemotherapy. So it's very interesting to
Be
see.
specific, right? Like, I mean, not all FGFs are created the same, right? So in cholangio, in our initial, because we published our initial 61 patients with infigratinib, and in that it was all FGFR alterations, and it was really the FGFR2 fusions that responded,
Right.
not mutations, and but not nearly as well, and not as robustly, and not as consistently. So the mutation, the amplification, I think those are, infigratinib has a cohort. Cohort 2 has other FGFR alterations outside of fusion. So, and it's only 20 patients. I don't know how much we're going to learn from it, but
at least this is a FGF1 to 3 intact, compared to pemigatinib, it was FGFR2 specific.
Right. It's curious that they're in cholangiocarcinoma, but apparently not in pancreatic adenocarcinomas.
Yeah, pancreas cancer really, the only ones I've seen in that situation are probably in the context of folks who have a high tumor mutation burden or MSI. They'll also have an alteration with in FGFR. I do want us to move over to our next few abstracts. This is a really engaging discussion. Thank you very much. I want to make sure we stay on time. If you can have the next slide, please. So we'll be going over 2 abstracts for hepatocellular carcinoma. Really been an exciting time for hepatocellular carcinoma after not having any drugs other than sorafenib for 10 years. The last couple of years have really been exciting. The first trial, can I have the next slide, please, is the IMbrave 150 study, which was a global randomized phase 3 study of atezolizumab and bevacizumab against sorafenib. So first study designed for superiority over sorafenib. And we knew the initial results at the main ASCO meeting earlier in summer, and here we were presented with the updated overall survival data. Just to summarize, the baseline characteristics were fairly comparable, average age of 64, and predominantly male population in the reflecting the disease that we're looking at. And what we're seeing here is an improvement not only in progression-free survival, but overall survival as well, with a median overall survival of 24 months versus 11 months with the atezolizumab versus bevacizumab versus atezobev versus sorafenib. Toxicities, really the toxicities that one would expect. The rates of grade 3 to 4 treatment-related toxicities were about 43% with atezobev and 46% with sorafenib. And then we'll go over one more study that was presented on the next slide, which was a combination of nivolumab and ipilimumab. So these are long-term results from the CheckMate 40 study, which was the multi-arm study that we've been seeing results trickle out over the last many years. In this study, they're comparing 3 different combination doses of nivolumab and ipilimumab. Nivo 1, Ipi 3, Nivo 3, Ipi 1, where they had 4 consolidative intense doses followed by Nivo maintenance. And then arm C was Nivo 3, IPI1 continued until progression. And really, in summary, the three arms are fairly well balanced. The things to look out for here, the overall response rate was numerically higher in, or fairly similar, I would say, between the three groups. But the overall survival in Nivo 1 IPI3 was 22 months versus 12 months with Nivo 3 IPI1 and 12 months in Nivo 3 IPI1 continued throughout. But that came at a cost. That came at a cost of toxicities, where any treatment-related adverse events with the initial more efficacious, apparently more efficacious arm, with a 27% treatment-related toxicity for Nivo 1 IPI3 compared to 15% and 17% in the other two. So two very exciting studies, one with combination of VEGF and IO, the other IO compounds. Probably gonna change the way we treat hepatocellular cancer, has already changed the way we treat hepatocellular cancer. ASCO came up with updated guidelines. Dr. Shroff, how do you synthesize all these new studies that are coming out, these and some other studies that are evolving for combination therapies for our HCC patients?
Thank you. Yeah, you know, I joke that when I was at my previous institution, I only did pancreas and biliary because all there was in HCC was sorafenib, and I was like, that's depressing. I'm not doing it. I mean, you know, it's bad if you think it's more depressing than pancreas cancer. And it's just been rapid fire in terms of drug development now. And I love the fact that, like, the biggest question on when you're trying to figure out what talk to give at ASCO and, you know, the ASCO Education Committee and all of these, we're thinking, how do we sequence? I mean, that's like a really great problem to have when it comes to hepatocellular carcinoma. So I totally agree. I think that my favorite part about the I Am Brave is that you don't, I say any sort of Kaplan-Meier that does not require a statistician to look at it is a fantastic Kaplan-Meier. So, you know, you take one look at that Kaplan-Meier and you know it's a positive study. I mean, it's dramatic, and it's completely changed the way that we're going to treat, or has already changed the way that we're treating this disease. You know, I think the things that kind of jump out at me is the things that we typically think about with immunotherapy, especially in HCC, is, you know, the durability. It's like that the curve is not dropping. You know, I mean, it's not. I mean, yes, it's dropping, but it's not dropping the way we traditionally think of as HCC patients that turn, unfortunately, and kind of quickly go south when they don't do well. So I think that's the most striking part about it to me, is there's a clinically meaningful PFS and OS that is long lasting. I think this study was well designed in the sense of it had representation from viral and non-viral etiologies. It was a global study with not necessarily an all Asian population, which oftentimes is a concern with HCC studies. You know, it was an advanced patient population between the amount of people who had macrovascular invasion and extrahepatic spread. So, I mean, it was, in a sense, in that sense, a real world population. Obviously, you know, when you're in clinic, the frustrating things are that we don't really see Child-Pugh that often. And, you know, that's the quote unquote criteria for every single one of these studies. You know, it would be nice to include maybe the B7s and things like that. And so it's a great study to have, and it's great results to have. It's a little trickier, I think, to implement, and it's not as generalizable as everyone wants it to be. The other thing that I always point out in this study is that everybody was mandated to have an endoscopy before because of bevacizumab, in terms of the risk of bleeding from varices. And again, you know, that's great. It's great that they were able to mandate it, but like in the real world setting, as COVID comes crashing down and elective surgeries and things like that are being, you know, held, it became a little tricky for me sometimes to be able to get an endoscopy, make sure the patient was cleared, and then start atezo-bev. And so it's, again, just like that real world translatability in what is a very sick patient population who have, you know, the complexity of comorbid liver disease and how you manage it. So, I mean, that to me, I think is the salient take homes. I think at the end of the day, we've seen this study kind of presented in multiple ways. We saw the primary endpoint presented as ESMO Asia, then we saw the PROs, then we saw the final OS data, and across the board, I think it clearly favors the atezo-bev arm in every single one of those readouts. I do think that there's something nice to be said about the PRO and the quality of life and the safety component. Again, obviously, if you're keeping it a child QA and you're excluding variceal bleeders, that changes the patient population a little bit. But in those patients, it was a really well-tolerated regimen, and we know that sorafenib sadly isn't. It's not the easiest drug, and it requires a lot of dose modifications and that sort of thing. The other thing I will say, again, just because for all of us, right, who start these patients on TKIs and that they have to have very close clinic follow-up in the beginning, and you're just trying to make sure that the cabozantinib isn't causing their hands and feet to fall off and all of those things. I mean, again, in the COVID era, having this kind of every 3-week regimen that's IV, and you're not worried about compliance, and you're not worried about how often you have to see them, et cetera. Again, I just think that in terms of clinical practice, it makes things a little bit easier, if you will. And then putting that into the context, I think the biggest issue now is, and I was saying this earlier, is now what do we do? Because there's so many frontline studies, including a nivo-ipi versus sorafenib study, and then there's the lenvatinib pembro, which, if you believe the initial data, looks really exciting. And then there's the cabozantinib atezolizumab, and then there's the durva-tremi. And there's all these studies that are done and waiting to read out, and they're all compared to sorafenib. And so I think we're going to end up, it's going to be like the pancreas story with the FOLFIRINOX and Gemnab paclitaxel, but I mean, with a little bit more complexity, because then we also have, what do you do next? And after IO, can you give more IO? And can you salvage an IO TKI with nivo-ipi, and should you? And all of these questions that I think, I guess, are good problems to have, because we're going to have a lot of, I think we're going to have at least a couple options, but I just don't know where to go now, which is frustrating a little bit. In terms of the nivo-ipi data, for nivo-ipi, I think, again, there's no doubt in my mind that in the people in whom it works, there is that usual kind of durable response that we see with immunotherapy, and that sort of the long-term follow-up that reiterates what the initial data shows is always really reassuring. And I think that median OS and the response and such are completely supportive of that. But I totally agree with you, Dr. Beg. I think it is toxic. 30% is no sizable number. I mean, it's no small number. It's a third of patients. And again, what I think is a pretty sick patient population at baseline. And so I think it's going to be tricky to figure out who the right patients are to receive this. When you look again, this study actually in the initial CheckMate 040, they actually had some B7s in there. And that was really, it was nice to see what nivo alone was like in B7s. I thought that was, again, kind of real world informative data. But here, again, this was very, this is a very pre-selected patient population. And I just wonder who we can give nivo-ipi to safely, especially when you have an option like what I Am Brave gives you. I think
it's a frightening regimen. What would you do for your average Child's B performance status 1 67-year-old HCC patient now?
Right now, I would probably give nivo. Yeah, or I mean because what's that? Yeah, I was gonna say or sorafenib. Like those to me are the ones that have some data. So, and it's just some.
Dr. Vijayvergia, Dr. Shroff talked about how the patients who ended up on this study were really cherry picked, right? They were scoping patients, they were excluding a lot of patients with Child's A. And in your experience, atezo-bev was FDA approved last year. What are we seeing, and Dr. Knopf, what are we seeing in the clinics in terms of what the trials are reporting and what we're seeing? Anything surprising with that combination for HCC?
All right, so you will hear a crying infant in the background, but hopefully my husband's got him, sorry. So yeah, so I think for atezo-bev, as Dr. Shroff mentioned, the cherry-picked population that was used in the study is hard to reproduce in clinical practice. You know, it's sometimes I've had patients like, you know, some patients who waiting on the endoscopy and deciding if I should do just single agent Nivo or with insurance approved, giving them atezo until I get the endoscopy done and add the bevacizumab after it versus just starting a TKI by itself. Which one is easier? Obviously TKI sounds much easier, like giving them some sort of TKI. So also the B7s and the sicker patients, at least because sorafenib has been around for 10 years, we have more experience and data to use that drug. So I really do feel I have struggled a little bit to use as a bev as much as I want to. The data is very promising and exciting, but trying to find the right patient where I can use this combination has been a real challenge to me in the clinical practice. So, and I think, I'm waiting for the pembrol and Vedneb data to show up, and maybe that would be a easier, I don't know, I'm just hope, we don't know. It's just all these different drugs that come in, but I think, it probably definitely need a regimen that is more easily administrable to patients.
Yeah.
Can I ask, is there any predictors for who's going to respond to PD-1 inhibitors in these cancers?
No, PD-1 expression repeatedly was not predictive. It's very rare for these folks to have actionable mutations. And one of the things which irks me about the HCC space is for decades we've been treating these patients on the basis of imaging and alpha-fetoprotein. We only started biopsying them. Trials were forcing us to do it, and they were being managed by non-oncologists oftentimes. And you see how adequate molecular characterization on diagnosis has revolutionized bladder cancer and cholangiocarcinoma and lung cancer. We're seeing that evolve right now, and I always feel the HCC space is suffering because of the lack of tissue or lack of well annotated repositories, because those folks with advanced disease were not molecularly characterized or just all characterized. So it is the chicken or the egg, but really right now we end up choosing our treatments based on what we think our patients can tolerate. I thought it was surprising, and Dr. Shroff highlighted how the 30% toxicity rate with ipi-nivo is really high. But with the early results of the durvalumab TREMI study, where they just had one loading dose of CTLA-4, they did see better early signs of activity, but not the toxicity. So it'll be really interesting to see how this evolves. I think we'll have to see how the bleeding episodes end up being a problem in the real world once we start giving these patients to, like our panelists described, where we may not have had the ability to scope them, or they had a scope in a couple of weeks ago as opposed to a couple of months ago, where we have to make real world decisions in treating them and see whether that's really a concern or not. Maybe that'll help sway us into identifying whether someone should get IO-IO combinations or IO-VEGF combinations or just the TKIs. And then what do you do next, right? There was a large data set analysis from the Hopkins group, which showed of the folks who start first-line therapy for HCC, only a third make it to second line. So the ASCO guidelines talk about, well, run the IO first and you can do your sorafenib and lenvatinib next. There's going to be a very steep drop-off at that point. I'm curious what other folks think about sequencing for this disease.
Agreed, completely. I think that is a concern. I always think about, and I had a trainee actually ask me this question in clinic the other day about, what to do with second line. And I was thinking it's hard. There are very few patients who get to second line. They get sick or something happens to them and there are some, they definitely do. And then you have the question, do you go to a TKI or which you would otherwise have done first line, lenvatinib, for example, or sorafenib, or do you do cabo, which actually has data after progression on a TKI? Because some may argue bevacizumab is VEGF targeted therapy. What is the benefit of more VEGF targeted therapy after you've progressed on one? There are different ideas and questions that come to mind. But in the lack of a better option right now. I think sequencing will become a very valid question that will need to be answered in this disease.
Yeah, I agree. And I think, the other question is also, is there a role for IO after IO, right? I mean, there's, if you're using IO up front, do you have any hope of using something later again? And, I mean, in theory, it would make sense that there's probably not a role for VEGF targeting or IO, but then, that's not always, again, like, the FGFR story says otherwise, right? There are other stories that say otherwise. And so you have to wonder, and I think that's the frustrating part for me about the trials that are ongoing right now, is they're not answering those questions that we're going to be faced with in clinic. They're all kind of racing to the frontline setting, and we're going to be, I think, left with more questions than answers.
Do you use the survival curves when you decide? I mean, I'd be very nervous about giving IPI at that dose to most of my HCC patients. But for a fit patient, 22 months overall survival is pretty impressive.
Yeah, I agree. I mean, but I agree. I think it's I mean, in clinic, it's going to be like this much of your patient population that can actually tolerate it. That's my concern.
The younger ones who have the fibrolamellar variant of HCC, the ones who are actually really good performance status and have everything good, there are different targets being studied there completely, right? That's like going in a different direction completely. So just further reduces the pool of patients where I could actually use this combination.
I wonder why in GI there's not predictors for PD-1, because in lung cancer, clearly we see smokers respond better, people who have a lot more neoantigens respond better. And so in a clinic like mine, where we see all sorts of cancers, we sort of take that thinking over to another type of cancer.
Well, upper GI, gastroesophageal malignancies have figured it out with the, I mean, it's not the cleanest, but with CPS score, and using that as a predictor. And then obviously in colorectal with the MSI high, but I mean, I agree. I think, and it's, I think the frustrating part about it is, even like MSI high, like nothing drives me crazier than when like my pancreas patient comes in and is like, oh, but I want the Keytruda. And I'm like, it's like less than 3% of pancreas that's MSI high, So, I mean, it's like the few markers that we know are valid is a very small percentage of the patients that we treat, unfortunately.
I will say the response rate endpoint is valuable. We heard in one of the talks I mean, we're talking about downstaging patients now. We're talking
Right.
about taking someone who may not have been resectable potentially for surgery. Or we're talking about, if there's vessel involvement, to see if we can help modify that so we can offer those patients surgeries. So I think a lot of progress in this space. Again, as Dr. Shroff said, back when we were training, this is not how we thought that it would unfold, and we thought pancreas may be ahead of the field, but clearly some tremendous progress that's been made in the hepatocellular cancer space, which is fantastic for our patients. So the last study that we'll be talking about is targeting pancreas cancer. So the Alliance preoperative modified FOLFIRINOX or FOLFIRINOX with hypofractionated radiation therapy for borderline resectable pancreas cancer study was reported. We knew of some of the results early on when one of the arms was closed for enrollment, but this is the first time we're hearing the actual results. So in this study, took about 120 patients and randomized them to receive chemo or chemo followed by radiation therapy. And then patients were taken for surgery and finished out their treatments with FOLFOX chemotherapy treatments. Average age for this group of patients was 62, 50-50 split on men and women, and about 80% were white. And when we look at our surgical rates, 32% in the FOLFIRINOX arm, and 32 patients in the FOLFIRINOX arm and 19 patients in the FOLFIRINOX and radiation arm underwent surgery. And of those who went to surgery, they saw an R0 resection rate of 88% with chemo and 74% with chemo plus radiation. There were 2 path CRs in the radiation group though. So the second arm with radiation was closed due to an interim analysis They didn't meet their endpoints, but the 18-month overall survival with chemo alone was 66 months with a resection rate of 49% and an AE rate of 57%, which is a little higher in the chemo RT arm. So Dr. Vijayvergia, how are these results impacting the care for our patients? What are your thoughts on this study in general, and how applicable it is for the patients who we see in clinic with early stage pancreas cancer, especially if you put it together with the SWOG 1505 study that was reported in the main ASCO meeting in June?
Yeah, thank you so much. Thank you, Shalaan, for the opportunity. So I think, this was a study that was designed for patients with borderline resectable pancreas cancer, so those who have some degree of vascular encasement, very clearly defined. I'm not going through it, but the role of pre-op radiation is a matter of debate, has been for a long time in this disease. And we think of this, the people who are against it say that, well, pancreas cancer is a systemic disease, and the potential for this local regional control that RT offers is sort of negated by the extent of systemic disease, and really it's not really helpful to do local regional control here. But we all know that patients with R0 resection do better, and hence local therapy, especially in the setting of vascular encasement, is like really appealing. So the Alliance 021501 clearly set out with a very nice goal of studying these options in our patient population. In this, they included only patients with borderline resectable disease, and they just compared the two arms. One had RT along with neoadjuvant chemotherapy, whereas the other one did not. The difference was the RT was a hyperfractionated, a short course, 5 days of radiation therapy, and in this setting, the arm 2 was closed early. So essentially, it set out to compare the two arms, but we really didn't get the results of the comparison because the second arm really closed pretty early for futility, which we're going to talk about in a second. But so essentially, it became like a single arm study of neoadjuvant chemotherapy in this disease. And we very clearly, very nice, 18-month overall survival of 66%, which was better than the 50% or 55% that they've read in studies. If you put SWOG 1505 into perspective here, that was a study of in patients with resectable cancer, so not the borderline resectable as our patients. These patients didn't have any vascular involvement, so they were very comfortable omitting radiation therapy in that setting, and it compared to different chemo regimens, FOLFIRINOX versus Gem/Abraxane, and in that their 2-year overall survival was about 44 and 47%. So again, coming back to the idea of a 66% 18-month OS with chemo alone in a borderline resectable population, it's pretty impressive. I think it's a good bar to set for future studies, and does point out that FOLFIRINOX in a neoadjuvant setting is effective. Another set of body of evidence supporting neoadjuvant approach in this disease. I think this is something that is becoming more and more the norm than actually just adjuvant. The Alliance study of neoadjuvant versus adjuvant FOLFIRINOX is going to hopefully seal the deal on that. But I think the bigger question here was whether the study answered the question of benefit of RT in this setting or not. And unfortunately, I feel the study, I don't think it failed. I just think the way it was designed and the way the results panned out was it just didn't answer that question for us, right? So they closed the RT arm because of lower rate of R0 resection, so they put like 37% as their rate, the futility margin, and the rate was about 35% in the first 30 patients on the RT arm. And but the patient had surgery within a week of completing their week to 2 weeks of completing their radiation. 2 weeks, I'm sorry. So there was not enough time after RT to say that it would have actually affected the rate of R0 resection. So I think so I really still struggle with the idea of that the closure was based on the rate of R0 resection, and for the benefit of RT, the effect of which was not really presented in such a short duration of interval between the completion of RT and the surgery. And there's a lot of other factors, right? Even the discussant today at the time of presented the idea of whether there was fibrosis associated with the radiation or the fiducials, which complicated the surgeon's assessment, especially in a study that's done across the nation and not just at high volume centers who, the surgeons may or may not be used to doing surgeries in post-RT setting. And that could have complicated their assessment and the ability to get R0 resection. We don't know. These are just things that we can hypothesize. Again, the other point of, there was 5 days of hyperfractionated radiation, but what exactly was that? Was it SBRT, IGRT, different types of it, because that can affect the relative efficacy. But I think so I really do feel that the role of radiation was not answered. Not so much there is no role for radiation. At least that's my opinion. Even though the study the study team and the discussant didn't think it that way, but that's just my personal take. Also, I think, in terms of toxicity, both arms are pretty, like 57 versus 63% rate of grade 3 toxicities, which is pretty much equal in the two arms, but we shouldn't compare them. But it's pretty high, right? There's this pretty high rate of grade 3 toxicities. And interestingly, only 20% in the RT arm and 30% in the chemo arm actually completed the entire treatment protocol, including adjuvant therapy. So it begets the question of, very few percentage of patients can actually get treatment after their surgery. So making neoadjuvant therapy more, more attractive in this setting, but also, do we need a better strategy overall that, more treatment is just neoadjuvant and nothing post-operatively, because most patients cannot get to it. I think I think what it means for me as a clinician is I think FOLFIRINOX is a very effective and a great standard neoadjuvant approach. Until 2 or 3 years ago, we had very limited data on the benefit of neoadjuvant therapy. We used to use it for a long time, but I think now we have enough data from SWOG 1505, the prior Alliance study 21101, and this one, that FOLFIRINOX is a good neoadjuvant approach. Unfortunately, as I said, I don't think the study answered the question of role of RT as well as I would have liked it to. There is, there are 2 studies, the PRIOPANC-2 and the PROTEGE. They're both European trials, the PROTEGE 44. They're both accruing. One of them is, using neoadjuvant FOLFIRINOX plus long course RT with followed by GEM adjuvantly or 5FU adjuvantly versus chemo alone. And then the PRIOPANC, which is the PRIOPANC-2, which is the PRIOPANC trial, which was for borderline resectable, where they got GEM RT and then surgery, compared to just neoadjuvant chemotherapy with FOLFIRINOX. So these are 2 important studies that we are waiting to see the results of, which I really hope can answer the question of role of RT. Now, I really do feel there is some role of RT in patients who have arterial involvement. There was about 40% of the patients on our study which required vascular reconstruction in this setting, and so I think that can be improved in local control. It can be achieved. It's a smaller, the incidence of local recurrence is smaller than distant disease, so we need a bigger sample size to see the benefit of local control in this setting. But that's my take on it.
That's a great observation. I think your observation that radiation across the board for everyone who's receiving preoperative therapy isn't the answer, and we need to find who those patients are. To Dr. Knopf's earlier comment for the HCC study, where we need to try and become a little more nuanced. We have 5 minutes remaining on our webinar. I was hoping that we could go around the table and let our panelists riff on what their major take-homes are. You guys suppose in your major thoughts, real 10,000-foot level. Dr. Knopf, you want to go first?
Well, for our practice, I think we need to look through our options for HCC. We see a lot of HCC, and so that's become much more interesting, but not a lot of patients at the clinical trials. So we need to sort of read the trials closely to see what sort of regimens would be appropriate for patients who have Child's B or performance status 1, which we usually see. The molecular analysis of GI cancers is much more interesting, and that's probably something that I would need to read a good review article on and think about a little bit more, and review also the upper GI stuff. And cholangiocarcinoma is much more interesting molecularly than it was 3 or 4 years ago. I did not pay attention to this study, and just as a weird comment, I think it's very bizarre to take patients to surgery just 1 or 2 weeks after radiation. We never do that in other tumors, and the radiation oncologists have handicapped themselves by having that protocol there. So I don't know that it would change our role of radiation in these malignancies, but it is sort of an interesting trial. So I we like to wait a little bit until the paper's out sometimes to make big decisions, because the molecules may not be available to us anyway. But certainly much more interesting and nuanced for GI cancers than it was 5 years ago.
Thank you very much. Dr. Vijayvergia, take homes from the GI Cancer Symposium.
Well, I think, as Dr. Knopf mentioned, I think all the buzz is in the hepatobiliary cancers right now. I think benefit of all the molecular targeted therapies. Also, I'm excited about the upper GI studies too. There are some good upper GI cancer studies that have come into existence. And then in terms of locally the pancreas world, I really do feel there's more studies in this neoadjuvant setting that I think tailoring and coming to that idea that we were talking about, Dr. Begg, earlier too, and the 60% toxicities, grade 3 toxicities with the regimen is extremely high. Only 20 or 30% of patients actually complete full treatment, and the average age of patients in this the study was about 63. The average age of diagnosis of pancreas cancer is 71. So in the real world, when you actually translate this into practice, I think it's high time we need a study for older folks and to tailor this treatment in some way that we use it, because where we are going with the standard of care is really not deliverable, because we don't want another atezobev to come up for the older patients, where most of those we see like that.
Absolutely. So lots of work in precision medicine with new combination therapies. Dr. Shroff, you want to close us out here?
Sure. I agree with all the panelists. I think, precision medicine is the name of the game for GI malignancies, both in upper GI now and hepatobiliary or biliary. And I think that's been proven. It was poetic that trastuzumab deruxtecan got FDA approval this weekend of all weekends. And so, it just furthers the fact that there's a role for targeted therapies in most GI malignancies. And I think that's my primary take home. I will say, just so you know, so the Alliance study, the protocol was written as 2 to 4 weeks. And for those of you who have not done a lot of SBRT, so MK, Matt Katz and I are very good friends. We've been colleagues for many, many years. We hate SBRT. That's my honest take home. And that's because it is impossible to operate on these tumors after about 4 weeks. It's very different than traditional 28 fraction 50.4 gray. And surgeons will say that to you time and time again. And there was QA here. So there was a radiation oncology, there were central radiation fields that were checked for QA. So my actual take home is I do not think SBRT plays a role. Now, whether or not regular course radiation plays a role in a subset of patients, I think my general takeaway from GI ASCO is that we can't treat everything with one specific approach, because I think targeted therapies tell you that. I think Bev-Atezo is not translatable for everybody, and I think RT is not for everybody. And I think that's, I would say, my general take home from what this weekend has shown me, is that we have to be thinking about every patient and nuance their care.
So precision medicine. So that's a great take home that kind of brings it all together. Thank you very much to all the panelists. Really appreciate your time. Dr. Gala, would you like to say a final few words? I hope that we see everybody together next year at the GI Cancer Symposium. Otherwise, we'll see you on Twitter. Dr. Gala.
Thank you so much. Thank you. This was, an amazing discussion. I really appreciate the time all of you took, and to all of our attendees, thank you for attending. We will be sharing a recording of this on LARVOL social media pages, so please check that out. And like Dr. Begg said, I really hope we can all meet in person next year. Thank you so much. Good night.